Morning, can you hear me? My name is Mark Tsuchida. I guess I would be called unofficially the lead programmer for µManager at UCSF. And I would like to talk to you about µManager. I really want to thank the organizers for giving me this wonderful opportunity to present µManager, because this is actually a really exciting time for us. We have been developing µManager at UCSF for a long time. But next month, we're moving the project to an independent company called Open Imaging, and I'll introduce that a little bit at the end of the slide. And just for the interest of full disclosure, I'm going to be the CEO of this company. So as the title says, µManager is about image acquisition. And so we all seem to share a microscope icon, but ImageJ, of course, is a general image visualization and analysis program. It's not limited to microscopes. µManager's role is really to produce the images, if you're doing microscopy, that you might be analyzing or visualizing in ImageJ. And so our job is to talk to hardware. We control microscopes. We control digital cameras to acquire images, and also all the components on microscopes, such as shutters, stages, filter wheels, turrets, lasers, switchers, all sorts of things like that. And we really view ourselves as the counterpart of ImageJ for acquisition. µManager is the only full-featured open source package that's really available for image acquisition in microscopy. And I think it's accurate to say that we share a common goal with ImageJ, which is to provide a common platform for scientists to use and to build upon, to develop methods on, and be able to share those methods with other scientists. µManager development started about 10 years ago. It's been a surprisingly long time. In Ranvel's lab at UCSF by Nico Sturman and Nenad Amodai. And since then, we've been getting a lot of code contributions. I think now there are about 100 authors in the code base. We are getting an increasingly large number of people adapting µManager for their experiments. These graphs are showing the registered users who provided their email address when they first started µManager. We have about 20,000 now. And we estimate we have about 9,000 computer systems that are running real microscope hardware worldwide. And presumably these are underestimates because you can opt out of reporting. We also have a very active mailing list and about 700 publications mentioned in µManager. A number of open source projects have built upon µManager. People from OpenPolscope and OpenSPIM and IC are here. Also outside of academia, the company ASI, which is a sponsor of this meeting as well, is developing the DICE-BIM scope, the dual inverted selective plane illumination microscope, originally developed at Harish Roff's lab, and their software for running this is completely based on µManager and open source. And so I've talked about open source. The other characteristics that we like to stress about µManager is that simple things are possible through an intuitive interface. So a basic image acquisition experiment is very easy to understand. But if that's not enough for you, you can script µManager. You can extend it in various ways that I'll talk about a little bit. µManager runs on multiple platforms. This is particularly a challenge for device control, but we run on Windows, Mac OS, and Linux. And the asterisk is there because, of course, depending on which drivers are available, you can control different hardware on different systems. So by far, Windows has the most device support. But we are not tied to specific hardware from specific companies. We can drive microscopes from all of the big microscope companies like Nikon, Olympus, and Zeiss, and others. We can control more than 40 scientific cameras, including the major ones, but also a lot of other ones. And we control lots of devices, stages, shutters, filter wheels, LEDs, lasers, spinning disks, photo-targeting modules, spatial light modulators, etc. And the full list is really becoming really large. And µManager provides a uniform interface for all these devices so that if you build a microscope or set up an experiment using, for example, an XY stage from company A, and you want to run it in a different lab with an XY stage from company B, then you can just transport your experiment and it'll just run. And so on the previous slide, I showed you all the devices we support. We have about 170 of these specific modules for each type of device that we support. And because each module might support a range of devices, we probably support hundreds or thousands of devices. And close to 100 people have contributed device adapter code. And this is in particular an area in which we've gotten a lot of code contributions, perhaps because it's modular and easy to understand how to extend, and because there are so many devices for microscopy. And so this has really been a success as an open source project. So I want to ask you, how many of you have used µManager? About a quarter. And how many of you have done scripting or programming that involves µManager? A few people. Great. So I would like to show you a pre-recorded demo of what the µManager user interface looks like. Find my mouse pointer. Okay, I'm going to restart that. Oops. There we go. Okay, so, oh, you know what? I'm going to switch to a different way of doing this because the pointer is not behaving the way I would like. So when you start up µManager, you see the familiar ImageJ toolbar. We ship with a bare copy of ImageJ 1 so far. Eventually, we want to make it seamlessly work with Fiji. And this is the main µManager window. The top part of it provides a sort of central control panel for your microscope with buttons and selectors for various hardware components. And I'll go through some of these in the demo. First thing I want to show you is getting a live video feed. Press the live button, you get a window displaying what your camera is imaging, and you'll see me manually focusing the microscope. This is a slide of stained mammalian kidney cells. µManager has some nice histogram-based intensity controls, so you can scale the intensity similar to the brightness control in ImageJ, but slightly nicer. And you can turn on auto stretch. And because the µManager windows are actually ImageJ windows, you can use the ImageJ tools to select a region. And in fact, the intensity auto scaling works with that region of interest. And for this demo, I'm going to turn on intensity scaling so that it ignores extreme bright and dark pixels. So I'm stopped live mode. And the next thing is this top right area of the main window. And each of these items in this table we call configuration groups. And these are combinations of settings on your microscopes that the user can set up. And you would typically set up these groups for each of the things that you frequently switch on a microscope. So one example of that would be the illumination wavelengths for fluorescence. And for example, we have a group called channel defined here, which is currently set to GFP. And that setting would reflect the position of multiple devices, such as filter wheels or maybe LED color or filter turrets. So I'm going to switch that from green GFP to cherry. And I can snap an image. And now you see the mitochondria instead of the actin stain. And I can switch that to another channel, DAPI, and you can now see the nuclei. And so that's sort of the interactive basics. And now I'm going to show you what we call multidimensional acquisition, which is how you would run an experiment and acquire data. And so the multidimensional acquisition window provides controls for time points, multiple stage positions, z-stacks, and channels. The first thing I'm going to show you is channels. If you turn on channels, it lets you select which channels you like, what exposures to use, and a number of other advanced things. If I click acquire, the microscope is going to take 3 images, 1 for each channel. And what's happened now in terms of real microscope control is that the microscope has moved the turrets and filter wheels into the correct position for the DAPI fluorescence, open the shutter, snapped an image, close the shutter, move the turrets and wheels to the position for green fluorescence, open the shutter, snapped an image, close the shutter. So that's the kind of control we're talking about. And so once we've acquired this image, we can use the histogram controls to scale each channel, turn back on auto stretch. You can display the channels individually. And switch back to composite mode. So going back to multidimensional acquisition, next I'm going to show you Z-stacks. If you turn on Z-stacks, it lets you set the start and step size of a stack. And currently this is set to relative Z, which means given your current focal plane, it's going to go from negative 10 microns to positive 10 microns in 5 micron steps. And then click acquire. Since channels are still enabled, it's going to do 3 channels at each Z slice and going through the 5 Z slices. And that's the 5 Z slices, and you can play back the result. And this is a little bit of an artificial example because this sample is way too thin at this magnification to actually do a Z stack. It's more like a defocus and focus stack, but you get the idea. Time points are pretty straightforward to understand. You can either do fast streaming acquisition from cameras, or you can do time-lapse acquisitions through the same interface. In this case, I'm setting up a 10-second interval time-lapse. At each interval, it's going to do the same Z-stack with channels that I've been showing you. So that's the first time point. And it waits until the 10-second time point, does the second time point. Waits, and does the third time point, and so forth. And finally, we can do multiple stage positions. And for simplicity, I'm going to turn off time and Z for now, but you can, of course, do all 4 axes, and you can also have autofocus mechanisms operating at the same time. So multiple positions is configured through this position list window. And one way to construct a position list is to turn on live mode and move around the stage and mark positions. So push the mark button, that's the first position. Move the stage manually, select another position. And another way to do this is to use the create grid button down there, which will allow you to create a regular rectangular grid, which we might use if you want to acquire a large region and stitch the images later, perhaps using one of the ImageJ plugins. And so now I'm acquiring that multiple position setup, and it's doing the 3 channels at each position, moving around the stage. And I just want to briefly talk about saving images. You can, of course, save images after you've acquired. That's just a standard save dialog. But perhaps if you're running a long experiment or even an overnight experiment, a better way is to enable saving images before you start the acquisition. And that can be done in the multidimensional acquisition window. You just check save images, give your image a file name and location. And if you start an acquisition, it'll save each frame as it acquires it so that you never lose data if your experiment goes wrong. And the window indicates that the image is already on disk. And if you open up the folder where these images were saved, you can see that µManager saves images as an OME TIFF file. But this is actually an OME TIFF file that also contains additional µManager specific metadata that records all of the state of the microscope as known to µManager for each frame. Okay. So I'm going to switch back to my slides. And so that was the basics of the main user interface of µManager. µManager also has plugins. So µManager is technically an ImageJ plugin, but it also has its own plugins. And those can do, among other things, things like running specialized acquisitions that cannot be covered by the standard multidimensional acquisition. It can control special devices like particular autofocus devices, spatial light modulators, laser targeting devices. And some of them can process images as they're acquired. So if you want to do a flat field correction of live images, that's possible now. I'm just going to highlight a few of these plugins. One of them is the HCS site generator, high content screening site generator plugin, which will allow you to configure your stage for a multi-well plate and generate a position list for your selected wells, which you can use to run a multidimensional acquisition. We have a particle localization and tracking plugin. This is more of a post-processing plugin, but it can do super resolution localization based on Gaussian fitting and other fittings. One of the fun ones is the live tracking plugin. This was developed in order to image fast-moving cells, and this movie is showing a fish epithelial keratocyte that moves at about 1 micron per second. So in a normal time-lapse experiment, it would quickly move out of the field of view. But what this plugin does is it uses image cross-correlation to compute how much the cell has moved and counter-steers the hardware stage so that the cell stays in the frame of view. And this has even been run for hours without losing the cells, in a lucky case. And the last plugin I'm going to talk about is called Slide Explorer. This is a very popular plugin, and I'm going to show you a pre-recorded demo for this. And so Slide Explorer is a way to quickly get an overview of your slide and find locations of interest. And when you start it up, it's going to initially snap a single image of your current field of view and display it at 1x. And then you can use the controls to zoom in and out. Here I'm zooming out. And when you zoom out enough that you see areas outside of the field of view, the µManager, the plugin will automatically steer the stage to that position and snap an image and fill in those tiles. So it works kind of like a Google Maps, except that you're actually controlling hardware to acquire the images. And in addition to zooming out, you can also pan around with the mouse. And it has a nice full screen mode. So this is the same mammalian kidney cell slide that I was showing. But if you have, for example, a slide with sparsely plated cells, you could use this to get an overview of where the cells of interest are. And then you can switch to navigation mode, and you can double-click to return to the positions of interest. Maybe do a more detailed acquisition there. Or you can use the ImageJ selection tools to select a whole region. And then, unfortunately, the bottom of this movie is cut off, but there's a button that creates a position list, which you can then use for multidimensional acquisition. Okay, so that was the µManager plugins, and there are a lot more of these. So if you're interested, you can check this out on our website. And these are the following few slides are not about plugins, but a couple of advanced aspects of µManager. One thing I want to talk about is the support we have for acquiring data at higher speeds. And so all the acquisitions I've been demoing here so far are run in software. And what that means is that the computer is sending out commands for each movement of the devices. So for example, if you're doing a multi-channel Z-stack, the computer will send the stage a command to move the Z position to a particular slice. it'll send a command to the illumination devices to switch to a particular wavelength. It'll turn on the illumination or open a shutter. It'll snap an image, turn off the illumination, switch wavelength, etc. And each of these commands have to be sent over whatever communication line, serial, USB, or other. And it depends on the interface, but in particular for serial devices, each of those commands can take several milliseconds. So that's quite a bit of overhead when you have a lot of devices in a complicated experiment. And also computer-based timing is jittery because a lot of things are going on a computer with just a few CPUs. So one way to overcome this is to use hardware-triggered sequencing, and that means connecting hardware devices directly to each other. One example of how that might work is to use scientific camera's TTL output line, and that would, if you put the camera in a streaming mode where it uses the camera's internal clock to accurately time successive frames, it'll send out, as it acquires images, 5-volt TTL pulses at each exposure. And you can feed those pulses into devices such as a piezo Z-stage or a fast illumination switcher such as LEDs or an AOTF controlling lasers. And if you pre-program these devices to cycle through a predefined list of positions, then when you start the camera, it's going to send pulses for each frame and the devices will advance to the next position. So you get the same effect as the multidimensional acquisition, except that there's no communication with the computer. So it's all very high speed and accurately timed. And another way that this can be done is using an Arduino board. Arduino boards are $35 hobbyist microcontroller boards, but we actually have a µManager specific firmware that you can load onto these boards. And you can use these to do the programming part of this hardware triggering, so that if you have an illumination device that can accept TTLs for different channels but doesn't have the programming capability, then you can use this. And these movies are just going to show you what sort of speed up you might get from this. And in this example, the experiment is a 4-channel time series with 10 time points. And on the left is going to be a view of the illumination in a normal software-controlled acquisition. And on the right is the hardware sequenced version. So that's the same number of frames, but you can see the speed up. In fact, you can't even see all the flashes in the movie on the right-hand side. And this can also be done for Z-stacks if you have a piezo stage that's, of course, faster than a motorized Z-stage. And this movie is showing an example of a Z-stack acquired with hardware triggering. And this movie is actually playing at the real time that the Z-stack was acquired. So you can see how fast you can acquire a Z-stack in multiple colors. And the nice thing about the way µManager does this is that once you've configured the microscope correctly, all the user needs to do is turn on or turn off the hardware-triggered sequencing, and the multidimensional acquisition interface will automatically decide which of the parts of the acquisition can be hardware sequenced, and it automatically load those sequences onto the hardware and run it. So it's very easy for users to figure out how to run fast acquisitions. And so the next thing I want to highlight is also about speed, but we've put quite a bit of effort into optimizing µManager for high data throughput. And as many of you know, the recent, maybe last several years, these CMOS cameras have been coming out with very large chip sizes and fast frame rates, and these can produce data at over 1 GB per second. And that's quite a bit of challenge to handle. But if you have a fast enough solid-state disk array, then µManager is capable of acquiring and writing data at about at over 1 GB per second. And the movie is just illustrating this with 800 FPS acquisition of a chytrid fungus flagellum. So I want to switch to extending µManager. It's open source, so you can do whatever you like with it, but we also have specific interfaces, of course, for extending µManager. And I already mentioned the fact that you can have modular device adapters to control new types of devices. And those are written in C++. But aside from that, you can also control µManager from several languages. The layer that sits above the device adapters in µManager is called core, and that's the layer that unifies the language you use to talk to devices. And that's also written in C++, but we have wrappers for Java and Python, so you can talk to it from C++, Java, Python, and Java environment scripting languages, as well as MATLAB. And the top application layer, which we call Studio, is written in Java, and it provides a Java interface for programming and scripting. And of course, you can also access everything in ImageJ. The standard way of scripting µManager is to use the built-in BeanShell scripting capability. And you can combine hardware control and image analysis through ImageJ facilities. And we have a built-in script editor. And people have done all sorts of things with this, but just a few examples. You can run an automatic exposure to determine the correct length of exposure that would require image analysis. People have done camera characterizations, determining the quantum efficiency. Automated screening experiments where you move around a stage and automatically determine which fields of view contain cells, for example. And also the scripting is, of course, a very good way of prototyping things that you might later want to turn into full plugins written in Java. We have extensive wiki documentation on our website, which is micro-manager.org. And we have a very active mailing list, as I mentioned at the beginning. One of the interesting facilities we have for remote collaboration, so to speak, is an automated troubleshooting feature, which we have built into the program. And this has really helped us. So what it does is if you start it and then do some steps that have issues maybe in hardware control, it captures all the communication with the hardware and other things that happen in µManager and allows us to debug device issues, even for devices we've never seen. So that's an interesting facility we have built in. So that's an overview of µManager as it currently stands. And I want to spend the next few slides telling you about the future of µManager. And the first thing is µManager 2.0. The current version is 1.4, 1.4.22, by the way. And 2.0 is an effort that's really being spearheaded by Chris Weisiger, my colleague, who is in the audience right here. And the major component that's updated is the user interface, and especially the image viewers. So in 2.0, we have a separate window, a floating window that displays the image histograms and metadata. And a new feature is that you can attach multiple display windows to a single data set. So a similar idea to what IC does. And in this screenshot, we have two displays showing different z-slices of the same data set. And you can even link displays so that you can animate a time series in two different channels simultaneously. Aside from the image viewer windows being completely reworked, 2.0 is also going to introduce a user profile system. This allows users to save preferences individually, even if they're using the same account on the operating system. And this is important for driving microscopes because multiple people typically use the same computer in such situations. 2.0 also offers a clean and much more object-oriented scripting API for manipulating data sets and displaying them. And it's currently in an alpha preview, and you can actually download alpha builds from our website now if you want to try it out. And we hope to release this later this year or early next year. Beyond 2.0, we have a number of ambitious projects that we're thinking about. And we're currently designing a modular and extensible acquisition engine to replace the current code that's behind the multidimensional acquisition. And this will allow more flexible control and also better customization by programming. We're going to add a more flexible image processing facility, which I'll talk about in the next slide. And we hope to better integrate with ImageJ 2. And for example, that processing pipeline might interact with ImageJ ops so that you can process images easily while acquiring them. And we hope to add a standard test suite for device adapter modules so that we can eliminate a lot of device-specific issues that we always run into. And so I said we want to have a flexible image pipeline. So basically how µManager works is that it runs an acquisition which spits out images, and those get placed into what we call a data store that can be either backed by memory or disk. And then we attach a viewer to that so that the user can see the data store. And µManager is really about the flow of images. It's a dynamic process to acquire images. And there are things that are really advantageous to be able to do in that same framework. And one of those is processing images as they are acquired, even in live view. And so we want to provide a image processing pipeline. And this is not as much a reinvention of various workflow systems like µIME as much as a very specific targeted thing that's optimized for real-time processing as you acquire images. And we already have in 2.0 the capability to attach multiple viewers to a single data set, but that could enable us to incorporate different types of viewers. So for example, we could have a 3D viewer dynamically displaying images as they're acquired. So maybe things like Clearview could be integrated with this, and real-time processing could do things like deconvolving 3D stacks on the fly. So this is going to provide building blocks for a much more diverse range of things that you might want to do in conjunction with image acquisition. And we hope that this will make it a lot easier to integrate µManager-based acquisition to downstream processing and analysis tasks. So I just want to summarize everything about µManager. So I hope I've convinced you that it has a simple interface for running basic experiments. And we have a lot of plugins and keep getting contributions of plugins for various advanced features. We have a lot of capabilities for extending µManager if you're a programmer or if you can write scripts. And we have a very strong community, and we're very happy about that. It would never have been possible for a small team to maintain, let alone write a program that can control thousands of devices. So that's, I think, it's a very big success. And we want that to continue. And it so turns out that our NIH grant is running out this fall. And we have made the decision to move the development of µManager to an independent organization in the form of a small company, which we've named Open Imaging. This is actually going to happen next month. So Chris and I are going to be leaving UCSF to start this. Our basic goal is not going to change. We want µManager to be a community-driven project. We want it to be open. We want it to be open source. And we hope that our collaborations with other open source projects will continue as they currently are. Open Imaging will be providing, obviously, open source for µManager, free downloads for every release version. And we are going to be providing services, including technical support to users primarily, and in addition, various support services to companies such as device manufacturers and vendors. And in particular, for users, we have what we call a user service program for µManager. So if you're a user or you know a µManager user, you might be interested in this. It's going to be an annual subscription program, including technical support and downloads of nightly builds. And we can also provide assistance for installation and configuration and specific support for microscope core facilities. And this is going to be a way for, I guess it's really a shifting in the funding model from government or grant funded to user funded. And so this is sort of going to be the primary means by which µManager development is going to be supported into the future. And we've been receiving a lot of positive feedback about this, and we are pretty confident that we can make this work with enough help from our users. So we are very excited about this. And you can learn more about this at the Open Imaging website, open-imaging.com. And so I want to end on the note that we really view ourselves as a counterpart of ImageJ in the area of image acquisition. And we firmly believe in the goal of providing a shared infrastructure for scientists, a common community-driven infrastructure for scientists to base their experiments upon and be able to share that. And that's really going to be the goal of open imaging as well. So we hope to continue that and are excited about future developments in ImageJ, which we want to incorporate as well. And with that, I'd like to thank a number of people. I want to thank Nico and Ron for letting me work on this and also to carry it forward in the future. Chris Weisiger is my colleague, he's here. And there's been so many people, I just can't list all the people who have contributed to µManager, but thank you all, and thank you for listening. Yes, Carl. So that's a little bit in flux. It's going to be hosted by Open Imaging, but we currently have a Subversion repository, and we really want to move this to Git. So the final form, after some transition period, we hope is going to be on GitHub, like any standard open source project. But we might go through some steps where Open Imaging hosts Subversion or something like that. Any other questions?
I'm trying to use µManager along with ImageJ to take an image, process it, control some other devices that aren't cameras or µManager control devices, and then continue the experiment with that. What would you say is the best way to go about it? I've been trying to make jar files in Eclipse, download them, and then have a single script that controls those things.
Yeah, I mean.
So how can I extend it to control other devices?
Yeah, that's sort of a difficult question because it probably depends on what exactly you're trying to do. So we can talk more later. It's about controlling devices outside of µManager simultaneously with a µManager experiment, for those of you who didn't hear the question. There are a number of ways to do that. You can extend the multidimensional acquisition, but that capability is currently limited. We are working on more of that, as I mentioned briefly. You can also just write a script to run your whole experiment in µManager and talk to other things, but I can talk to you later about more details. Yes? Yes. Yeah, we have a plugin called Intelligent Acquisition that provides sort of a framework for doing that using ImageJ macros. Yeah, that should do it if you can do it from ImageJ macros. And of course, otherwise, you could also run a script that does that. It's called Intelligent Acquisition, yes. Back there? That is an interesting idea, and yeah, we should talk about that.
I mean, we have the µManager wiki, of course, which has all of the information that anyone has ever written down about µManager, and then there's the community mailing list. Some work towards merging that wiki with the ImageJ website as a whole. Yeah.
Yeah. Any other questions? Okay, thank you very much.